Good afternoon. Let's start slowly so then we can go over it's a long lecture today. So I will see if I can put here, it's not so loud. I like to have it in my hand, but maybe it's too loud. So I am Mariana De Lorenzo. I'm associate professor in the cell biology and molecular medicine department. Here is my email. So try to do it before the exam, like few days, so if you have questions, you can email me, and then we can make an appointment. So we can go over the question by Zoom, or if you are in the school, in my office. And then also I am in the Office of Education for the medical school. And then some of you, I meet you because I am academic advisor with Dr. Larry Gasper. So today we are going to go, I need to get used to it. Okay. So here I divided the class in different objectives. And you will see the dynamic of the class is like I'm going to show you some things are highlighted. Doesn't mean that only the yellow is the whatever you need to study, but the content, I will make you clear what is the important on each slide. So I will go over, and then if you have questions, you can stop me. And the same when the people are in the Zoom, please stop, say something in the word so I cannot be seeing the chat and also the slides. So if you have questions, you can stop me. And then also I divided every several slides. I did another slide with all the content that we review it. So then you see what are the more important concepts. So the principal objectives will be like to identify the different mechanisms in cancer, also to study the important role of the tumor microenvironment and angiogenesis in the tumor progression. We will see understanding the multi-hit model of cancer and what is the theory and what is the reasoning behind that. To analyze the genetic basis of cancer, and also we will see some examples of oncogenic mutations and also tumor suppressor mutations. So cancer is like is a complex, they call complex multi-step disease, is associated with different accumulation of multiple DNA mutations. So we are going to see that people sometimes they misunderstand and they say, oh, they all of a sudden they have cancer. So cancer is the malignant disease that is caused by the accumulation of different mutations along the life. So then what happens is that this mutation will lead to some, what we label here, the regulation in the cell proliferation, in also the differentiation, also we will see later in apoptosis. So we are going to see what are all the hallmarks of cancer and how these are deregulating based on different mutations in different genes. So what happens is the main concept is that you have DNA damage, and what happened, the system in the organism, we have different, what we have here, detoxification and repair systems. But what happens if those systems fail? So that DNA damage, it will pass to to the daughter cells. So what happens is that DNA damage can be occur due to different exogenous genotoxic compounds. We are going to see at the end the examples of different carcinogens. So could be radiation, chemical compounds, tobacco smoke. We are going to see as an example how a lot of compounds are carcinogenic in the tobacco smoke, different virus, or then also some chemicals that there are in the preservation. That is why nowadays everybody say that eating food that they are processed, because the processing has some chemical compounds, right? So all these things, they can affect the cells having mutations. So then based on that, if those mutations cannot be repaired, like we say, we have a system like detoxification and repair system. If that system fails, that DNA damage, it will pass to the progeny, right? In the other cells that they are the daughters, they will have the DNA damage. And that will have chances. We will see how is what they call the clonability of the cancer. So what happens is that if those mutations are in these three more important group of genes, those are the ones that they are very important for the cell proliferation and also apoptosis. So what happens is that if they are in these critical genes, we have three groups of genes. We call oncogenes, tumor suppressors, and also caretaker genes. We are going to see those in the next slide. What happened? What are the characteristics of cancer? So cancer cells are characterized, and here is a summary of all the characteristics that they have, the cells. First, they proliferate without need of extracellular signal or stimulus. I always tell the student we are going to see the receptors and the signaling. So what happened is kind of like, thank you. It's kind of like a short circuit, right? So then you have continuous signaling that the cell has to divide and proliferate, but then you don't have any extracellular stimulus. That is because there are a dysregulation in some part. It could be a receptor, it could be the ligand. We are going to see that. Also, the cells that cancer cell ignore all the signaling that they restrict growth and proliferation. Ignore the signal for promote program cell death. For those that they saw in biology previously, so apoptosis or the cell program cell death is all happening when you have a cell that has a DNA damage. We have the system that can be fixed, right? That cell can die by apoptosis or can get arrested, so then they don't proceed proliferation. In this case, in the cancer cells, they ignore those signals. So the cell damage, the cell that has the mutation, it can keep going through the proliferation and have daughter that has also the mutation. Also, there are modifications between the cell-cell interaction and the cell extracellular matrix interaction. We are going to see some tumor cells, they have abnormal shape and size, and also abnormal ploidy. So do you know what is ploidy? Is when the cells they separate, right? They have the segregation of the chromosomes. In this case, like the things fail, one daughter will have sometimes they have more amount of chromosome than another cell. So that is a different ploidy, referred to the different number of chromosomes to the normal cells. Also, they have promoting growth of blood vessels. We are going to see this is called, the procedure is called angiogenesis. And then on top of that, the cells that there are cancer cells, they can invade surrounding tissues and metastasize to a target organ in a distance, right? They have, in general, they go by the bloodstream to another organ. So these are all the characteristics of the cancer cells. So to make a summary, how these cancer cells can happen. So what happened is that we mentioned already the carcinogens, right? And then what happened also, we can have in some cases, a disbalance on certain hormones, and then also some exposure to some virus that we are going to see. They can have some oncogenes that they can carry with their genome. So what happened in synthesis, that cells will have a genetic damage, and then that damage, it will lead to some excessive growth, excessive proliferation, what we call the transformation of the cell. This is another way to say the oncogenesis is the process of development of cancer. And again, we say that this is genetic. When the people say it's a genetic disease, is because we're talking about it happens at the level of the DNA, right? So not all the misconcept is that they can be hereditary. No, not all the tumors are through the heredity, and we are going to see the examples of those cases. In this case, we have due to some disbalance on the genetics or the environment, because we say that can be some chemical compounds like carcinogens that there are in the environment. So there are a DNA damage in one somatic cell, and that somatic cells, what happens is that it start proliferating of a clone, and then additional mutation will happen. Those additional mutation will lead, as I say, faster proliferation or growth resistant against cell death. That will lead to a tumor and then metastasize. So we are going to see all these different points in the next slides. So I mentioned that if the mutations happen in these three groups of genes, protocol gene, tumor suppressor, and caretaker gene, is when you have These categories, do you have the influence of carcinogens? This is the definition of carcinogens. For those that they don't know, substance found in the environment or radiation that increase the risk of mutations in these 3 different categories. So these are when I explain you things to you. I used to do that also. When I learn something, then you can explain to people that they are not having this concept, right? So do you see that some people say that we have already the cancer on ourselves, right? They say that, oh, the cancer is inside the body. No, what we have it is what they call proton genes. Are these genes that they have a function in the normal organism? So that function, if they have a mutation in there, then there are no more a normal regulator, right? So once there is a mutation, it's called oncogene. So originally it's a proto-oncogene. So when they have a mutation, the name is oncogene. And what happens is an excessive activity or gain of function mutation. In generally, when you have a mutation on a proto-oncogene, it leads to oncogene. That protein is called oncoprotein. And then what happened is a gain of function mutation. And in the tumor suppressor, and in generally, the proto-oncogenes are involved in all whatever are their receptors, signal transduction, all these molecules that are very important for the cell proliferation. And what happened, the other group is the tumor suppressor. Tumor suppressors are more involved on the restriction of the cell cycle. And then when they become inactive, right, or they call loss of function mutation, there is when they are proceeding to have a cancer, right? That cell proceed to have cancer. For example, P10 or, P10 or TGF-beta, we are going to see the example P53. So they are all molecules that they are what they call, for those that they saw already the cell cycle, they call the checkpoints. So like there are restriction points where the cells pause and the molecules signaling in a way that if there are a mistake or there are DNA damage in the reparation, so what happens is that cell will stop there. It will die by apoptosis or will stay what they call in arrested state. So then they don't keep proliferation. But if there is a mutation in this tumor suppressor, there are no more restriction, right? So the cell keep proliferating, even though it's not the right way, because that gene is mutated, so it's not working. The signaling is not working. That happened with P53, retinoblastoma molecule. We are going to see those examples. And the caretaker genes are part of this group of genes that they are in charge of this, check the integrity of the genome against the DNA damage. But imagine if the role of these genes is produce protein, and those proteins are involved in the control of the DNA integrity, and they are failing. So it means that if they are having what the cell has the DNA damage, that DNA damage will proceed to the different daughter cells. So these are the examples where the 3 categories that is very important, that if we have mutations there, it will lead to a cancer cell. So then another important concept that I want you to know is that we have it, people talk about tumors. So all tumors are not cancer. So you have it, what they call benign tumors and malignant tumors. So malignant tumors are cancer cells, right? What is the difference between these things? Well, so what happens is that you can have some tiny growth of a tumor, but if they remain locally, they have low proliferation rate. So what happens is, in this case, the tumor cells keep the same phenotype and differentiation state of the cells around the tissue. And also, they keep some cell-cell addition, right? So molecules that maintain the same ones that there are within that tissue. So it means those characteristics are, even though this is a tumor, right? You have a mass, it grows a mass, but if it has this characteristic, is the design benign tumor. Examples are the birthmark, warts, right? So then we have benign tumors, and then we have the malignant tumors. The malignant tumors are those ones that they already have some capability to invade the tissue of origin and the surrounding tissues. So they have a way to spread and create new tumors. How they spread is because they have secretion of certain molecules that they call proteases that they can degrade the extracellular matrix and they can move and migrate to another area. So if you have a question that they tell you all the tumors are cancer, so that is why you will have to say in the exam is false, because there have to be, but all the malignant tumors are cancer, right? And these are why, because they have this capacity to be able to invade the tissue, spread to secrete, to create new tumors, and then metastasize. As an example, I put the slides. It's very simplified here, but you can see it could be like, this is a gland, but it could be like a mammary gland, for example. Or, so you have this organization where you have, this is a specialized, the yellow is a specialized extracellular matrix. It's called basal lamina. So what happens is like every epithelial, in generally today we are going to talk, only in few slides are going to talk about leukemia and lymphoma. But then in generally, we are going to refer to all the solid tumors, right? So in this case, you have an epithelial, right? This is organized epithelial. If you see the cells has different shape where you have what they call the basal and the apical side. And then in the middle, you have a lumen, right? So this is a normal duct, right? Could be a duct of the mammary gland. But in this case, you have a benign tumor. You see how this tumor has the characteristic and stay in situ, right? So they didn't invade another area. But in this case, you have a malignant tumor because you can see in the paper, in the book, they color different color because secrete different molecules, the proteases I mentioned. So what happens is that they can degrade this area, pass through this epithelial normal, and then start invading in another areas. So this is the difference between the malignant and the benign tumor. And here you have an example how it's different. The epithelial is in this, now is in this representation. So you have one in red, you have one cell, that cell has accidental production of one mutation, right? So what happened is that cell has the daughters. So you can see how passing the different cycles, what happens is it could happen a second mutation. And those ones, they be accumulating, they keep proliferating, and you can have a third mutation. So that is why they say they come in a clonability, because from one cell, right, that got the DNA damage, and that DNA damage was not fixed, it passed to the progeny, and then you have first like a clone, and then you have the whole origin from that original cell. So that this is a malignant tumor because this still benign, but then with more dangerous cell proliferation, it start invading. And this next one I want to show you, let me see, well, we go through some definition previous, okay. So this is just to show you a group. I don't pretend you to, remember all kind of the details, adenocarcinoma, divided, but this is for your information. And then what I want you to know is the carcinomas, there are tumors that derivated from epithelial cells. Then you have the sarcomas, derived from the mesoderm. Then you have leukemias, what I say, we will not talk leukemias and leukemias and lymphomas. They are derivating from the cells, like for example, here is the white blood cells in the bone marrow, and these are from the lymphocyte and the plasma cells in the lymph nodes. And then you have the blastomas are very specific. They lost differentiation like little. So these are characterized because when you read the paper and you want to know what we are talking about, is the different type of divided of cancer, the type of cancers. Then I don't want you to remember the specific each of the tumors, but from where they derivate, at least so you have some knowledge after this class. So this is an example how when you are when a person has an operation, or when they do it, in this case, it's a pap smear. So this is a gynecological study. So what happens is they take a sample, and then they will be doing what they call a smear, and then they will study. They will stain with this characteristic stain. It's not a immunohistochemistry. This is simple stain. And I don't know, how many of you know what is this stain? What? H&E staining. Perfect. Okay. So for those that don't know, H&E staining is a simple staining that they do it in the pathology. Sometimes if they are in an operation room and they are doing some resection of a tumor, they take a sample immediately. The person one of the assistant run to pathology. Pathology can stain quickly and see what's going on and if they need to resect more or not. This is what happens in the big operations, right? So, but in this case, as I say, it's a smear. So then patients go to the doctor, they give this sample, and then they will do this staining. H&E stain by hematoxylin and eosin. It's very simple, but it allows to the person, like a pathologist, that they are very well known and trained for recognize structures. They can see where is a normal or non-normal structure. In this case, I show you how to, you will see like the hematoxylin stain, it has affinity for negative charge, so in general stain the nuclei, where the DNAs and the RNA, all the negative charges are there. And then the eosin is the one that is staining pink, more pink, and then is more avid for positive charge and then stain the cytoplasma. So in this case, we can see how in this, consider that these are different patients, right? So then this is a sample how this patient has a normal epithelium. They will report it because you have the organization. You can see how here very organized the line, like we saw it in the slide before, right? When I show you the line of the epithelium and you have all the nuclei very organized. And here you have a lot of cells like they could be fibroblastos. This one, the one that you have in the middle, a white area, that is a capillary or vein. So you have to see the characteristic that the endothelial cells are all around this luminal part, right? So this is a very normal epithelium, and this is the connective tissue. Connective tissue is all the extracellular, what we call extracellular matrix, but it's very well organized to give the content to the tissue, to the epithelium, sorry. So in this case, this second patient, they have it what they call low-grade intraepithelial neoplasia. So what is this one? What do you characterize for what we see before? Is a benign tumor. So this and this is still benign tumor. This is high-grade intraepithelial would report because you see how here you start having this abnormal growth, but still in situ, right? It didn't invade this. But you see how it start changing this extracellular or connective tissue, right? So here you have more, less pink, and you have more dark that could be immunocells that they are already invading that area because it's kind of like it's not more normal. Because we are going to see, it's very important, the tumor microenvironment, sorry, the tumor microenvironment, right? So then even though these cells didn't invade, they are secreting different molecules that they secreted the normal tissue. So this is still a benign tumor. This is benign tumor, but high proliferative rate. And this is definitely is like a malignant tumor because it's invasive, right? And then you see almost no pink, right? So then probably the cells, the fibroblasts, everything, they are what in this class, we don't have to go over all these details because we don't have time. But also what they say is that the fibroblasts that they are associated to they call cancer-associated fibroblasts or CAF. So they secrete different proteins that what they are secreting these fibroblasts here that they are normal. So that is why you see that different, all this also is full of a small nucleus. We see the dot in violet, there are a lot of immunocells that are infiltrated there. So that is an example how you can see it in the pathology, how you can see a normal tumor, a benign tumor, a normal tissue, a benign tumor, and a malignant tumor. Any questions still here? So now we are going to go more to the molecular part. So then we have examples like in 2000, and then in 2000 I was doing my thesis, and then I was working in angiogenesis also on tumor metastasis. So what happened is this group of faculties, they came with a very good review, and they characterized the whole of cancer. all they characterize the whole of cancer are all these substantial proliferative signal, then evading the growth suppressor signals, very, like we say, inducing angiogenesis, because the angiogenesis is very important for the tumor growth, activating invasion and metastasis, all these characteristics resisting to cell death. In 2000, that was the panel that they showed. But then in 2001, they came with a review with more characteristic, one more hallmarks. And these are the ones that I circled here. It was more complicated, this graph, and it has all the target different drugs that they are using in those years, but we don't care about the drugs. We care about the pathways. So nowadays is no more that in 2000, they know more that tumor promoting inflammation is very important. Also, they know about the important role of the immune system, how they avoid the tumor secrete molecules that they try to hide from the immune system. Also, it's very well known now that even though the tumors prefer glucose for the metabolism because it's fast to metabolize, but also, they have different pathways. So then that is why the dysregulation cellular energetics. So all these are the characteristics that they have, the hallmark of cancer, that they have supporting the point that from initiation of one cell that has a tumor, then they promote and then they can be progressed to the metastasis. And that is a summary of how important is the tumor microenvironment, I say before, because the cell-cell interaction, it makes signalings that they affect the tumor, and then the tumor secrete molecules that affect the cell-cell interaction. That context give all the support that for another group, these are there for those that they want to read the paper. This is a very big paper, but they came up with what they call meta hallmarks of aging and cancer, because we are going to see how in the majority of tumors that we have, solid tumors, age make a big role, right? So then what happens is, because we will see how we said before, one mutation is not immediately a tumor. So we had to have accumulation of different mutations. So then with aging, is more chances that people, of course, unfortunately, we have events that they are younger. So for those that I will explain you, we were in a conference a few years ago, and one guy from MIT is Dr. Robert Weiner, very famous. And then he was asked, why if in older people we have more risk, right? Because you have more age to happen all these mutations. Why in young kids that they have lymphomas or leukemias, it happen in the early age? So unfortunately, still the science cannot see how in these individuals very fast occur all these mutations that in another type of tumor like solid tumor, it happened in many years, right? So, but as I say, we are not going to refer to those in particular, but I want to mention to you. So the question is how these young kids has these tumors in the blood, right? Because they are most likely are leukemias or lymphomas. So then this paper is very interesting because they show how all the things that they can be changing with age, it will affect in the same, what they call very similar to a specific cancer hallmarks. So there are some interactions and there are some pathways that run in the same way. So then if you fix one of the pathways, benefit for aging and attack cancer or vice versa. So we are going to see, just only to show you and to start thinking why we say that it's nice to have a healthy aging, because that will reduce the possibility to have many disease, right? Because we have this is chronic disease that they associate with what they call age-related disease. Well, for example, we have the Alzheimer's, cardiovascular disease, cancer, right? So based on all this, there are different pathways. I don't want you to memorize, but I want you to understand, right? So those pathways that if they are dysregulated, it will lead to a more faster aging, and then probably risk factor for another disease like all this chronic disease. This is the paper that they were talking about difference and similitude between cancer and aging. And in particular, like we saw some nutrient sensing the regulation, it can happen in aging, and also what will happen in cancer, right? So then what happens is that they are proposing that there are some interventions, like for example, I was studying actually the caloric restriction in mouse models and how it's affecting on the metastasis. And cancer is very well known already for years, but nobody show about metastasis. So I was working on that. Also, if some people now they are doing intermittent fasting, and then some nutrients that they can mimic good metabolism for then help it with a good aging. So good aging, it will lead to less disease, chronic disease, basically. So no need to know the pathways, but I want you to know how everything is interconnected. And then they came with this cartoon that, if you have a good therapeutic intervention, will help it not only with aging, also to be more less exposed to several things that they can cause an early aging and then diseases related, and one of them is cancer. So I'm going to introduce you how this tumor microenvironment is more related to this. So for example, the ladies that we buy cream for the wrinkles, and this based on what is that? Do you know? Why we want to have the skin, we put creams that they activate what? The collagen, right? So on that line, I want to show like the extracellular matrix molecules, they are outside of the cell, right? And then they are part of this connective tissue. In terms of the tissue, we don't talk extracellular matrix, we call connective tissue. But in reality, are all these molecules, laminin, type 4 collagen, some of them they have type 1 collagen, fibrillar collagen, different molecules that are part of this extracellular or connective tissue. How this organized? Well, I show you very briefly the epithelium, how it's organized. So we have always they have an orientation, these epithelial cells, and then you have the basal lamina that we say, and then you see here the different, not only you have molecules like the collagen or fibronectin, or here we have hyaluronic acid, proteoglycan, laminin, we have also different cells, right? That they are not part of the epithelial or the tumor. They are mast cells, there are immunocells like macrophage. So then you have the capillary that I show you in the previous slides from the pathology. So all these, and then the important is the fibroblast. So all these are the support that they are supporting the connective tissue, that they support the epithelial cells, the epithelium. So then each cell, like I say, is organized. You have a apical side, and then you have the basal side in the epithelium, and then you have many connections between cells to cell. This is an example what I show you for the normal, the benign tumors. So we say that they keep this kind of a cell-cell interaction or cell extracellular matrix interaction like the normal tissue, right? So then when you have it, malignant tumors, that is no more there. So they are more lax and then can produce proteases that they degrade those connections, and then they can move the cell. So, and the cells also, not only in the tissue, if you have cancer cell in one of these epithelial, they have one tumor cell, not only in that context, they are surrounded by tumor microenvironment. They are, when they are traveling through the bloodstream, also they go with different associated cells, right? So you can see here the endothelial cells, and then they have the, I say, cancer-associated fibroblasts. They are already different from the fibroblasts that they were in the normal tissue because they produce different cells. They produce different molecules, and the tumors reply secreting different molecules too. The same happened with the macrophage, what they call tumor-associating macrophage. And this is an example from another paper that I talk about the dysregulation that they happen in the stroma, or what they call stromal is the connective tissue, and what happened with the age microenvironment. You have many molecules that there are no more normal in that microenvironment. So then they could drive tumorigenesis and progression. Let's see this picture where you have a healthy microenvironment, right? So you have senescence, you have apoptosis, you have here some apoptotic premalignant cell, right? So they have this premalignant cell means that has some DNA damage, but like they are in a context that is normal and healthy young tumor microenvironment is a microenvironment. So that cell will solve. So then you don't have a tumor or proliferation. But what happens when you have an age healthy microenvironment? It's still healthy, but look at here. You have more cells premalignant, right? Because the conditions here change, right? You don't have more, you have some already cells that there are here in the basal membrane. They are not tumor cells. These are what they call fibroblasts. So then they are changing this microenvironment, right? They propose is an age-healthy microenvironment. And here already you have some growing that is there are already tumor cells, right? So the tumor microenvironment here is completely different. You have different cells that there are, the proliferation of more cells. It brings more angiogenesis. It can be, so all this is a base of how healthy or not is some tissue. So when sometimes one mutation is happening in one context, it can be solved, like in this context, right? So it can solve and then die by apoptosis, that cell. If the environment is not the proper because it's aging and the collagen fibers are not the same, many components will not support that tumor cell. So then that tumor cell can become more malignant and then become a tumor and metastasize. So, and this is how we can see, this is a paper that was a study different molecules, but I want to show you here how these are a specialized way to see that they call the two photons. So then you can see the density of the density or the thickness, they call thickness of the layer of collagen, right? So here is the normal young one, and here you can see how many cells are invading there, and the collagen is not the same thickness that in the normal one. And in the case of these are a mouse, and they do it also like a composition of a two-photon microscopy, how here you have the composition of collagen, normal, young, and these are age one. So they are more less thick and the fibers are thinner, right? So this is just to tell you how important is the context of the microenvironment and how, because in the past they think about only mutations and what happened in the epithelial cell, but that is not isolated, right? And on top of that, not only in the humans, or if we study in mouse model, only the tumor is not only that tumor microenvironment and that tumor cells. You have to see that there are different organs. For example, I'm working on the fibroblast growth factor that is secreted by the liver. So how everything is connected, and then we need to start, because in the past they used only to study the mutations that happen in the cell. But now it's everything, it has a context, right? And it depends what they call the seed and soil theory, right? For the metastasis. It doesn't, so if you put a seed in a soil that is not proper, it will not grow, right? But if you put the seed in one place where it's proper, all the conditions are given, so the cell or the tumor in this case will grow. So that is, it's more focused on the metastasis, but the metastasis is a secondary growth after the primary tumors. Okay, so we talk a lot. Do you have questions about this? Okay, so what we are going to talk about now is the angiogenesis. Angiogenesis is one process that, now I'm being hot here, it depends. Today I was very hot or cold, depends on the moment. So angiogenesis, when the tumor starts growing, so what happens is they say approximately 1 million cells and the growth of 2 millimeter diameter. So what happens is those cells, they will need supply of oxygen, supply of nutrients, because in the middle, those cells are going to become dying by hypoxia, right? So you have lower oxygen tension in that middle of the tumor. So if the tumor want to grow, because you can imagine always the tumor is kind of like a little monster, right? That want to grow, grow, grow. So what happens is that it needs supplies. So that supplies is they will induce the formation of new vessels. And that are they call the angiogenesis, neoangiogenesis, neovascularization, angiogenesis. So what happens is that the main purpose for that is the promote of growth of the tumor by providing more oxygen, and then also open the path for tumor metastasis. Because the tumor, when invade different, it will go towards where a capillary. So where they localize a capillary, then they will go through the bloodstream. We are going to see that process. So the mechanistic of angiogenesis, what happened is that the same tumor secrete molecules, as I said, different molecules. Some of these molecules are basic fibroblast growth factor, tumor growth factor, and then BGF. BGF is the key one because it's vascular endothelial growth factor. So those factors, the tumor secrete that, and what happens is that it will induce the proliferation of the cells, and then because require oxygen. If they don't have oxygen, we see that then later we are going to see in this topic is that they can do glucose anaerobic metabolism. So what happens is that the production of BGF is stimulated by the hypoxia. When you have a lower oxygen tension is hypoxia, and that induces a transcription factor. The transcription factor is HIF1. HIF1 alpha in general, hypoxia inducible factor. That inducible factor, it will stimulate, right? That more production of the molecule. And what happened also, that HIF1 alpha, it will regulate and activate all the pathway that the cells, in the meantime, they can do this glucose anaerobic metabolism. Why? Because there are lower oxygen tension at that moment, right? So increases the chances of the survival in a hypoxia condition. So what happens is that then those tumor cells, as I say, secrete because they are invading, right, the surrounded area, then they reach a capillary, and then they will have to penetrate the vessel wall, what they call intravasation. So they enter in the vessel, they go through the bloodstream, and then when they reach the target organ, they will extravasate. So they leave the blood vessel and then enter in the could be a breast cancer, it could be a metastasize in the lungs, or some of them they do it in the bone. So what happens is that the cells will be migrating towards finding the capillary, and that migration, why I put this in green? I want to make a clarification, because one thing is the migration that the cells is doing, but the migration inside the cell, how is that? Is the reorganization of the actin, right? So remember, the reorganization of the actin cytoskeleton is the possibility to reorganize and migrate. But on top of that, the cells secrete different proteases that they go and then hydrolyze all the collagen or the laminin or whatever is in the extracellular matrix. So there are two different things. One is the movement per se that is originated by the reorganization of actin inside the cell, but then also open the path are secretion of the different proteases that they can open the extracellular matrix, and the migration can happen. And the cells will be moving towards nutrients or the oxygen, right? That is why they're looking for the capillary. So what happened is that, as I say, so one, they enter in the blood, in the wall of the vessel, then they go through the bloodstream, then they have to other in the target organ again, and then leave the blood vessel, enter in the target organ. So this is very stochastic procedure. They say that less than 1 in 10,000 cells exceeding the primary tumor will generate a metastasis. And then we are going to see sometimes those metastases, what happens is that they can stay in the organ, in the target organ, dormant, or they can grow, and that is why the patients, mostly they have the problem is when they have metastasis, because the metastasis, the primary tumor, they can reject it and remove it, but then sometimes the metastases are difficult to localize till they start growing. So this is a process, angiogenesis, it happened normally, right? But what happened in the normal organism, this is a process that can be, tissue cells, or they can be tumor cells. The reality is that when you have high oxygen, right, you have low this transcription factor, right? But when you have low oxygen tension, the hypoxia induce this high if one, and then they will secrete more vascular endothelial growth factor. So those cells, in this case, could be a tissue or a tumor, those cells secrete BGF, and then look what happened. This is a regular blood vessel, but look what happened. They stimulate those cells, and those cells, they start producing this capillary sprout. So then in another class that I did I do it for the PhD students, we can explain not all the vessels, the blood vessels, sorry, the endothelial cells has the same capacity. Some of them, they are the ones that they are producing this elongation, and another ones are the ones that they doing this spreading. But for the purpose of this class, you can see how a stimulation for these secretion molecules that they affected the capillaries, and then they produce this. And what they do is at the end, they try to cover the tumor. So then they get all the supply. And as I say, angiogenesis, it happen. If you have a wound healing, right? So you have a wound, it will happen that you have a process, and sometimes you need to have some angiogenesis to fix the tissue, but that will be regulated. In a normal process, some molecules will stop producing, and then that's it, no more angiogenesis. In this case, no, it's continuously, because the tumor grow more angiogenesis need. And this is the whole process that I was mentioning by part, right? So we saw it, a normal epithelium, then you have a one cell, give it to another two daughters, and then you have a benign tumor, then become a malignant tumor after several mutations. Then you have to have the intravasation, so enter in the bloodstream and go to a target organ. In this case, then the cell, they have to escape the blood vessels, so they have to extravasation, they call, enter in the organ. And sometimes they have these micrometastasis, and sometimes stay like this, small metastasis for years, but sometimes they just grow and they make a full-blown metastasis, right? That is the one that affects the patient. That is an example how you can see this is a gross, what they call gross pathology or gross anatomy. You have, these are the liver, right? And then you have different, these are the metastases. You see how they can be seen by eye directly. So what happens is that if you make a staining and then you cut this and then you make a staining with the H&E, so you can see how here you will see big cells that has more cytoplasma, and then big nuclei. And then here you have the tumor cells. These are the normal hepatocytes that we have it in the liver, right? But here you have very small nuclei, more violet, and you can see these cells are, even though you are not a pathology, you can see these cells are different than these cells. These are structures, what they call the canaliculus, the structure of the hepatocyte. These are completely disorganized. They look like different, like could be like a metastasis of the breast cancer or another type of tumors, right? So that is to show you how also you can see it with a microscopy. And these white things are in generally, these are fat cells, so probably could be like some kind of cirrhotic liver in that patient. So we have a lot of topics now. You have a question? Question? No. Okay. I thought you were moving the hand. So what we have it now, we are going to make a summary, right? We discussed the difference between benign and malignant tumors. We say that cancer is an age-related disease. Then also we discussed how important the proton gene tumor suppressor and caretaker genes, because there is where, if you have mutations there, you can live to have a tumor. The hallmark of cancer, the importance of tumor microenvironment, and then tumor angiogenesis and metastasis, right? So you have some question for this? We can pass to the next step. I don't know, you want to keep going and then so we leave early, or you want to have a break in half an hour? What do you prefer? Leave early, okay. I run the shuttles also, leave early. Okay, so let's go to the next step. So I mentioned before the multi-hit model of cancer. What is that? The multi-hit model of cancer stipulates that cancer cells originate from several successive genetic mutations, right? So we say not only one mutation lead to cancer, so you have accumulation of different mutations. So in this case, we say, like, if you have a first mutation in one cell, sometimes it could be a stem cell, but it could be like somatic cell, gives some cell growth advantage, right? So that cell produced like a clone. We saw it from the slide that we saw the clone. So that progeny that has the clone develop a second mutation. So what happened is that clone grow faster, resist to apoptosis, and become a benign tumor. So to see that thing, I don't know how many or how much in depth you saw the replication, all this process that the DNA, right? So the replication has these proofreading enzymes that they control. So imagine, I want to make you think about that. So if you have a system that it review, right? So there are DNA damage or not, and if that fail one time, and the cycle is still being faster, right? So it can make it more mistakes, right? Because you already have some mistake that make the machinery to make it not be able to repair in time. The cycle keep going, the cells keep dividing, and this sometimes there could be more mistakes. So if you see from that point, you see that there are more mutations, and the more mutation happen is more severe the situation. So then you have a third mutation that accelerate growth, penetrate in adjacent tissues, and then also start producing proteases. The proteases, for those that they don't know, they are proteins that they have activity to cleavage and other proteins, right? So then proteases, they are enzymes. So then the fourth mutation definitely will produce more proteases and metastasize. So some examples are, for example, we are going to see in the next slide, but a mole can become a melanoma, right? And then intestine polyp can become an adenocarcinoma of the colon. So that is why it's so important. Nowadays, we have more technology, more conscious to say the people to go to the doctor. But if somebody is feeling no good and they don't go to the doctor, what happens is that, for example, if you're at a certain age, they recommend to have a colonoscopy, right? So, but for those people that has a family history of somebody has a colon cancer, they make them do it earlier in age, at 35 or 30, I think now. So what happens is that if they can do that study and pick up the polyp, they can remove that polyp. It will avoid to many years form a full adenocarcinoma. So that is why it's so important, the cancer prevention, right? There are different campaigns that they are producing, like people, like the ladies who have to go to the OBGYN, the guys has to go to the doctor for the prostate cancer. So with age, it's very important and to follow up this. And that is the basic for what? And this is an example of this is Dr. Borges published many years ago, but it's a classic paper that they describe all the mutagenesis that happen, that it will lead it to invasion and metastasis, right? So I don't want you to remind which is first or which is less, but you will see that it's a normal epithelium, and this is a normal colon, and this is a colon that you can see, this is a colorectal carcinoma, a malignant, completely malignant. So what happened is different at the different levels. First, there is a loss of APC. APC is a tumor suppressor. What we say about the tumor suppressor is a loss of function, right? So the tumor suppressors they are there to control the cycle and restrain the cycle if there are some mistake in the DNA damage, right? So what happens is that if that is lost, that activity, the cell will keep going with the damage. So then start becoming hyperplastic epithelium, then you have early adenoma. So if that person has here a colonoscopy and they remove this, will be most likely some tiny come another adenoma, but most likely it will be stopping that this cannot happen, right? So what happened is that person didn't go to the doctor and stay and nothing happened. So then they have more activate, you can have activation of KRAS. KRAS is an RAS is an oncogene, proton oncogene. So there are many cancers that they have mutations in RAS. Bless you. Then it happens that they have also other tumor. I think that we are, I think it's very humid today because I was always warm, but today I am cold warm. So what happened is that it happened more mutations, and at the end you have an invasive and metastatic tumor. So why they call multi-hit of cancer? Because you need several mutations to happen this. So this is a very characteristic for the colon cancer. They did it for another cancer, but this was one of the first pioneer description. Doesn't mean that all the patients that have cancer, it happens, exactly first this one. Maybe in another one, first is the P53, right? But what happened is they did a big study with a lot of patients, and they did all these molecular characteristics. So what happened is you have different mutations that the main point is different mutations will lead you to have that. If the polyp or the adenoma is not removed, then will happen that tumor. So the multi-hit model of cancer is supported, right? Experimentally supported by different things. For example, you can see that all the cells in a human tumor have inactivated the same X chromosome, so supporting the concept of the clonability, right? They're coming from the same original one cells. Also, the incidence of cancer, we discussed in certain way the other before, right? So incidence of cancer increases with age. So we discussed that, right? Because we have more time to accumulate it, more mutations. Also, another characteristic is that if a risk factor of a risk of esophageal cancer is increased slightly in people who drink alcohol, right? As well as in people who smoke. But it's increased more in people that do both things, right? What happens is that you have the exposure of different carcinogens. And then on top of that, the alcohol expose different carcinogens and provide cumulative mutations. The alcohol, you remember, I don't know if you saw this in the school, but you have it in the liver is where we detoxify, right? So we are going to see at the end the carcinogens. So if you drink alcohol and then you have all this, sometimes they use it the same, what they call the cytochrome P450. There are molecules that there are the enzymes to detoxify toxins, right? So that it will bring it. So a person that has risk factor for esophagus and drink, but if there is a person that has risk factor for esophageal cancer and then drink and smoke, it has increased the risk. So that is why I say that tobacco smoke, right? Don't then say, Dr. De Lorenzo say, if you smoke, you will have, it happens, I always give the same example, that it happens that you are being exposed to, I think it was 300 to 400 toxics, carcinogens in the tobacco smoke. So it's an exposure. Nobody, some example in one of my friends in Argentina, the mother was heavy smoker and also she was drinking. And then all the family complaining. I remember when we studied, oh, mama, this is smell terrible. Why? So she smoked, and then 10 years later, the father has lung cancer. Who died? The father. She's still alive and she's still fine. So it means that, for her to be exposed to those chemicals, nothing happened. But if he has some risk factor or some, sometimes they call a hot sequence in the chromosome. We don't know because they didn't do all the details, but the reality that the one that died from cancer is the husband because he was exposed to secondhand smoke, which people don't believe that is true, but yes, it's true, right? So in your family, if there are somebody smoking, and in this case, what happened is the one affected was the husband. So it's not that I say everybody that smoke will have it, but it's a risk factor. We say we are exposed to different chemicals. So what happened is that also another experimental approach that they can be supporting this is that in the cultures, when you have it oncogene MYC, right? The proto-oncogene overexpresses oncogene MYC affect the proliferation and the apoptosis, but oncogene RAS lead to non-proliferation and absence of the apoptosis, right? Or senescence. So what happens is that if you have transformation that cell with the two oncogenes, so automatically lead to the transformation because you are putting more pressure on that cell, right? To happen more the transformation. Another example is like what they said, progression in testicular cancer. In some way, we see this in the cancer, in the colon cancer slide. What happened is why we have endoscopies and biopsies, we say, right, that if you lost the tumor suppressor APC, then the cell leads to the formation of polyps. But if you can, on top of that, you have a mutation in RAS, that polyp become benign tumor. But then also if you have another mutation, it becomes a malignant carcinoma. These are all examples how they can be adding different mutations and how you can be increase the risk of the formation of tumors. And this is in a graph where you have these three group of, we say the three group of genes. And then in this case, we have these genes that control cell growth and cell proliferation. So this is simplify the picture that I mentioned before, right? So in this case, you have RAS promote the growth, but not the proliferation, right? So then you have MYC that promote proliferation, but not the growth. And what happens if you have, these are tumors, free tumor mice, right? So there are mice that they have genetically modified MYC. So they will live, right? Here is the age and days. They will live, right, till the almost 200 days. But what happens is that if you have this colony of mice, they have both mutations. They have MYC and RAS. You will see 150, they will be all the mice dead, right? They will be tumor free, but they will be 0. So it means they all die. So why? Because they have expression of the two oncogenes. So then the proto-oncogene, we talk about proto-oncogene. In this case, we talk about oncogene. So when a oncogene represents this derivated from the proto-oncogene by mutations. When is that happening? When it happens? So we say that the proto-oncogene becomes oncogene with a mutation. It happens in the DNA sequence that lead to production of proteins that is hyperactive. Remember, we say gain of function, right? Is a hyperactive protein called gain of function mutation. In that case, it will be called oncoprotein, right? So one example is RAS. RAS is in, I mentioned it before, right? So RAS is in many tumors is mutated. So it's one of the characteristic proto-oncogenes that they become oncogene. So these are examples how you will have it, a proto-oncogene converted or reactive as a converted into an oncogene. So for example, it will happen once you have a deletion or point mutation in a coding sequence, right? So what will happen is that mutation will lead to an hyperactive protein made by in normal amounts. So it's an hyperactive, so will enhance the proliferation, but the amounts are normal. In the other cases, when you have a regulatory mutation, the mutation happen in one of the regulation areas, so the normal protein greatly overproduce. So you have more of that protein, normal. Another example is the gene amplification. So what happened is normal protein greatly overproduced, right? So they are copies of the same gene. And also will happen on the chromosome rearrangement. When they are chromosome rearranged, it can happen two things. It can happen that the change happen nearby regulation, the DNA sequence cause normal protein to be overproduced. Why is that? Because it's under a promoter that is overproducing, right? Because the rearrangement was dysregulated. And then what happened is we are going to see a example later. So it will be the rearrangement will lead to fusion of active transcript genes producing hyperactive fusion protein. That will be a fusion protein that didn't exist originally. Right now that they are oncogene, they call oncoprotein or fusion protein because it didn't exist before. We are going to see later an example. So in summary, what we explained before, we have here number 1 and number 2, they say the first two lead to synthesis of new protein, right? And the last scenarios in 3 and 4, the protein coaching is produced in an excessive amount. So first is producing a synthesis of new protein, the other is the different amounts. So the number 1, we say the point mutation, a single amino acid substitute makes the protein hyperactive. Then you have it, the chromosomal translocation with fusion, that we say that we are going to see the example, the BCR-ABL, because that protein didn't exist, it's a new protein. Then the chromosomal translocation with new promoter. That is a very classic example. So for example, what happened in this, in the lymphomas? This is mentioned, we say no tumor solid, right? But in the lymphomas. So the lymphoma, the lymphocytes, right? If you see the immunosystem, the lymphocytes, they produce immunoglobulins, right? So that is the main point. So the main role is that. So then imagine that the immunoglobulins promoter is very strong because they need to produce a lot of immunoglobulins. So when you have a translocation in this cell that unfortunately has a chromosomal translocation, so MYC, that we say that is a proton coaching, MYC is being put under the control of the immunoglobulin promoter. That is very strong, right? So then now, instead of producing the immunoglobulin, they also start producing MYC, dysregulated, right? So that it will lead to an abnormal growth, right? Because this is not the regular way that, the MYC doesn't need to be hyperstimulated when it's not needed. So that is one example of the chromosomal translocation with new promoter. Or the other one that we saw it is the amplification of the protein coaching translation, but increase of the copy, the increase of the fragment in different areas of the DNA in the chromosome. So those are the examples how proton coaching can become oncogenes. So summary of the virus, because I mentioned in one of the first slides that virus can be sometimes oncogenic. So what happened is that there are, basically what I want you to know from this slide, that there are two different types of virus, right, that they can be affected. One is the retrovirus. So for those that they don't know, the retrovirus, they are RNA genome and is reverse transcripted to DNA. That DNA is being inserted in the eukaryotic genome, right? So like they have RNA, they cannot insert directly to the DNA, right? Let's say that is a human and they get one virus, that DNA cannot be. So what happens is they have enzyme that is called retrotranscriptase. So that reverse transcribed into DNA and the DNA inserted. So there are two ways. One is when the virus proliferate and incorporate a proton coaching from the host, so then into this own genome. So the virus enter, they reverse transcript the DNA into the DNA enter, and then when the virus proliferate, they have the proton coaching in their genome, right? Further mutation, it will lead to the formation of the oncogene, but that was already in the DNA, in the sequence of the virus. So then another way is the virus genome is insert close to the proto-oncogene. So then what happens is that the stimulation of that production of the proto-oncogene now is oncogene, right? So this is a characteristic of the retrovirus. Two things important. One is how they are RNA genome and they need to be transcribed in order to enter in the genome. And there are two ways. One is when they incorporate the proto-oncogene from the host, and the other is when the virus enter in the genome closer to a proto-oncogene, so then this regulate that and become an oncogene. That is some examples, but then also we have the DNA virus. The DNA virus, they already has, as the name say, the genome is a DNA, so they don't need to be retrotranscribed. And then what happens is that they include oncogenes and incorporate them in the host DNA. So like it's very easy to enter in the genome and then leave. When they leave, they can take some oncogenes, right? So they go infecting another cell or another organism, and then they will be able to produce a permanent transformation, because when they insert in the DNA, they have this oncogene. And some of the examples are the human papillomavirus. So that is, I want you to learn this procedure. Don't need to memorize the name of the virus, but you need to know what are the two types of DNAs, the virus, and what are the mechanistics. And this is a table for those that they are curious, all the names that if you want to know the names. One of these very characteristic, we have the RNA retrovirus, right? The HIV is a retrovirus. And then here you have the hepatitis C. So these are the names for your curiosity. So we talk about virus. So why not? I included a few years ago this slide, because bacteria also, they say that they have some of them, they have some participation, right, in tumors. So what it means that, to give you some concept, we don't have time to enter in all the details, but the concept is that the bacteria, mostly they are limited to cancers that they are associated with the GI tract, right? So all the part of and sometimes the lungs, but there are basically analysis they done by oral, fecal, or tissue samples. So in particular, the number one characteristic is that Helicobacter pylori is associated with problems that the humans group of one, they are considered a carcinoma, even though it's not a molecule, it's a bacteria, but it's associated, very strong associated with what they call in generally gastric tumors, right? So then H. pylori, none of the bacteria associated in human cancer has been formally proven to be a human, except of that, right? No other bacteria, only the H. pylori, it was characterized that is mainly human carcinogen. So what happened is that, so you can also study, there are some studies that they are done in colon carcinogenesis, but this H. pylori is the number one that is characterized. And these are some examples, how some stomach cancer related to the H. pylori, colorectal carcinoma, some people associate these enzymes, you don't need to memorize. H. pylori, yes, because H. pylori is very well known. The other ones are characterized, but they are seeing that mostly what happened, why the bacteria are associated with a different type of, and generally these are what I say, GI tract, right? So it's where we talk about the microbiota, because we always talk about how good is the microbiota. Okay, so let's talk about in what conditions they can be good or bad. H. pylori start growing abnormally, and that is why affected the humans that they have H. pylori. Sometimes they give a treatment with antibiotic, it solve the problem, that's it. But sometimes if they don't go to the doctor and don't follow up, that because what happened is that, I don't know if it's this one, no, but I will tell you later. This one is a show, you don't need to memorize this also, but look, this is exactly the hallmark of cancer, right? So in this paper, some scientists start showing how different bacteria's dysregulation affect some of the hallmark of cancer, right? So you will see some of them, they have what they call augmenting the hallmark and then inhibit the hallmark. Basically, that is what I want to show you, that some bacteria dysregulation can be on favor of the hallmark, so increase the hallmark characteristic of cancer, and some of them inhibit. You don't need to memorize because actually this is a paper, but it's not 100%, in all the tumors the same. So this is something that they propose. So the important point from this is, one, we say that H. pylori is one of the bacteria that was even considered human carcinogen. And the other thing is that the concept that they affecting the dysregulation of bacteria can affect the hallmark of cancer. And then we have two options, that microbiota, it can have anti-tumoral effect. That is why we talk about that if we have healthy microbiota, the anti-tumoral effect of the gut microbiota. So everything that make it secrete molecules capable to fight tumor, prevent tumorogenesis. But what happens when these different bacterial induced mechanisms, so when there are some what they call dysbiosis, right? And dysregulation of bacteria in our organism. So what happens is that sometimes that gut dysregulating microbiota can have pro-tumoral effects. And they are associated to be more like kind of is associated that secrete toxins. So those toxins will interfere with the host cell growth, with the layer of in the case if it's the gut, you have the layer of the cells on the gut, they get kind of like inflammation, erosion, and that is why. So if you have a dysbiosis, what they call in the bacterial growth, normally, and then you are not follow up with the doctor, sometimes this can be dysregulated. That is coming to also something that we say about that one of the hallmarks of cancer, the later on, the one that they published in 2011, is inflammation, right? All the inflammation that is chronic dysregulated in certain areas, not always, but in certain areas, like for example, the GI tract, it could cause problems later on in age, right? Because you have a erosion of this area of the layer of the epithelium. So we saw it in this second part. We saw multi-hit model of cancer, genetic synergy. Then we have how the proton coaching converting into coaching, role of the virus, role of the bacteria, and previously also we saw it the tumor angiogenesis and metastasis. So now we are going to see, we saw how the oncogenes that become proton coaching become oncogene. In this case, we are going to focus on the tumor suppressor. Remember, the tumor suppressors are, in generally, the role are associated with the checkpoints in the cell cycle and inhibit cell growth and proliferation. So the mutations that block the activity of tumor suppressor, remember, being very important, they cause loss of function mutation. So what happened? If you have the majority of the type of tumor suppressor are inhibitor of the cell cycle, we say recently, receptor of cell signal transducer that inhibit cell proliferation, checkpoint proteins to stop the cell cycle if the DNA is damaged, then also activator of apoptosis, and then DNA repair mechanistics. So these are generally the tumor suppressor. For those that they don't have familiarity with the cell cycle, remember that we have the cycle, we have different gaps. Gap 1 is to enter the cell get prepared to enter in the synthesis of DNA phase. Then you have the gap 2 that prepare the cells to get into the mitosis, right? So there are three characteristic checkpoints. We have checkpoints entering S. Why is that? Because the cell start checking if the environment is favorable and every condition is in place. So then for proceeding to the cell cycle. I think they have, sorry, I see that there are one question in the chat, I think. It is supposed to be of an oncogene from oncogene. No, the proton gene is the original, and when they have the mutation, it becomes oncogene. That is why I mentioned, I mean to say. Okay? So then another thing, the checkpoint, the second checkpoint is the checkpoint that is closer to start the mitosis. Why is that? Because now the cell has to check not only the environment, also they need to check the DNA is replicated, right? Because then we are going to go to the division of the two different cells. And the other one is the one in the middle of the mitosis, they call metaphase checkpoint, is the cell has to check with different signalings, right? So that all the chromosomes are attached to the spindle. Because remember, the chromosomes, they get some with the microtubules, right? That they connected to the pole. So they will have to be tracked in the after the metaphase, anaphase, and then the telophase. So what happened is that all these three areas are supported for different tumor suppressor molecules that there are where they are, if they have mutations, it will not be able to make this check. So what happened, checkpoint control timing, monitor DNA synthesis accuracy, and ensure correct chromosome segregation. That is the role of the tumor suppressor. Oh, why is not moving this? Okay, great. Okay, let's go with this one. I don't know, maybe no more. So what happened is that another example, we say that retinoblastoma is RV. Retinoblastoma control the cell cycle is one of the tumor suppressors, right? We have P53 RB. This is retinoblastoma. Retinoblastoma, we will go see what are the scenes. It's very important for the control. So we are going to see here. The problem is, I don't know if this one is this working. It doesn't pass by this working. So then in a non-proliferative cell, you have that in the cell cycle, if you remember, you have the cyclin-dependent kinase, CDKs, and we have the cyclins. So what happened is that you need this complex to be able to phosphorylate different proteins, right? So what happened is that you have P50, P16. In generally, the tumor suppressors are all P16, P19, P15, right? So they name like this. But in generally, this molecule is being inhibitor of the CDK, right? So they are complex like this. So what happened is this is inactive because you have this inhibitor. So what happened is that complex, CDK cyclin D, cannot phosphorylate the retinoblastoma. So retinoblastoma is associated to the E2F. E2F is a transcription factor that stimulates the transcription and the expression of the S-phase genes, right? So then the R44B is there. Is actually in the picture doesn't show, but it has few sites phosphorylated. But here you have a lot of sites phosphorylated, right? I think it's two here and 5 sites here. So what happened is that in a condition where the cell is under proliferation and all the stimulus is inducing the proliferation, so this guy, the P16 get inactive. So release the CDK. So the CDK can complex, right? So this complex phosphorylate more. So what they call hyperphosphorylate. This is hypophosphorylative RB. And this is hyperphosphorylative RB. So what happened, RB is inactive, so cannot complex with E2F. So now E2F is a transcription factor and stimulate the synthesis of all these, right? The expression of the S-phase genes. So now you have the proceeding of the cell cycle. So imagine if retinoblastoma, RB, is so important and is mutated or is deleted, this will not happen. This is this control, right? So the cell will keep going always to the cell proliferation, because there is no restriction. So this is how it works, the retinoblastoma molecule. Another molecule, the tumor suppressor, is P53. P53 is mutated in half, it's like RAS. We say RAS is mutated in almost a lot of cancer, also the same. What is the function, main function of P53? P53 is a tumor suppressor, and also what happens is that is censoring, right? If there is DNA damage, here is by, if you know you are in the sun a lot, or if you smoke a lot, these are like a graphic that they show in the slide. So the DNA damage, what happens is that P53 get activated. So the function is that is a signaling pathway, but okay, it get activated. When it get activated, it will transduce the signaling to different molecules to do, they have three options. One option is to arrest the cell, so it means that the cell will be in gap 1 or gap 2 and will not proceed with the cell cycle. Two is activate the DNA repair system, so the DNA repair will fix that DNA damage. But if fail, in the normal situation, if it's failing, the cells can die by apoptosis, that cell, because that cell has a DNA damage, so it's not better to have it. But that is if everything is fine, working fine. Imagine when P53 is mutated or deleted in the cancers. So the cell will not follow this, and it will keep going through the cycle. So that is another example how a tumor suppressor, right? It can get mutated, affect the progression of the tumor. So now an example is like, this is a tumor, like we mentioned before with the mice that the tumor, bear mice, tumor that they are free of tumors. Mice that they are free of tumor. So this is survival. These are three different colonies of mice. In this colony of mice, they have P53 normal. Look, the animals, they live, right? There are 500 that still 100% of the colony is alive. In this case, you have this mouse colony, they have heterozygous P53. You see already start dying some animals. So at 500, you have less. You have only survival of, let's say, 70%, right? But for those mice that they have manipulated, genetically manipulated, and they have mutated, deleted the P53, right? Minus P53. So there are no P53. They die at 250. All the mice, they die. So then that you show you how affecting the P53, it will induce the tumor, because they die because they have so many tumors that the animal die. So then I mentioned another thing that I put it here in the tumor suppressor area, because there are some things that, we say that some tumors, when they metastasize, they have dormancy. The metastasis, it reached the target organ, it grows a few, so sometimes the cells in normal condition will they will follow cell death, but what happens is that sometimes they have only one cell that they are in the target organ, so they call dormant a solitary cell. The cell will get arrest between the gap 0 and gap 1, and then they will not proceed with growing. In another cases, they form some growing, and then they get that mass of cells, they get in that site micrometastasis for many years. And in another way, what happened is that they call that they can be some rewake, so then they can grow a full site metastasis. So those are tumor cells that can, these are the characteristics, right? The cells, they die, so that will be the best thing. Then they can enter state quiescent on dormancy in one single cell, or micrometastasis, or proliferate to grow in more micrometastases. So those characteristics are based on you don't need to be exactly expert in dormancy, but I want you to introduce that concept, because you see sometimes people say that, oh, so-and-so, the family relative, they have cancer, and then for many years nothing happened, and then all of a sudden it felt, and the tumor grow. So what happened is, it shows in some opportunities, what happened is that when a patient or a person has for many years survival and didn't have metastasis, but it has these dormant cells, right? So what happens is when the person has an accident or an injury, it metabolizes a lot of molecules from the bone marrow, interleukins, inflammatory cells, and those things produce molecules that they can reawake, what we call it in cancer, reawake of the metastasis. That is what happened. It's not that the injury make the tumor grow. It's kind of like all the metabolites and different molecules produce interleukin, inflammation molecule, all this, it make stimulate that metastasis that was dormant. That is the explanation. That is why I want to introduce the concept. Hmm? So then another concept that I want to introduce is what they say, what they call loss of heterozygosity. I will give you an example, but the situation is this. That is inherited predisposition to cancer. This is what they call why some families have higher prevalence of cancer. Okay, what happened is that if there is a loss of function mutation in both alleles, right? So then you have that, what they call higher probabilities of developing cancer. And then also what they call from the perspective of the genetics is loss of heterozygosity. What means that? For example, retinoblastoma. Retinoblastoma, that protein is characteristic from some tumor that is called retinoblastoma. It happened in the retina, right? So what happened is that RB, as we say, block the shift one phase in the cycle. So if the person has RB positive negative, is heterozygous, right? So what happened is that if happen one mutation more, is loss of heterozygosity. Okay, we are going to see the example next. Another one is APC. Check that all these are tumor suppressors, right? So prevent the expression of the protococcine MYC. So what happened is that APC predisposed to colon cancer. And then BRCA1, block the cell cycle progression, BRCA1 plus minus predisposed to breast cancer. Somebody knows in the famous, how they call it, the shed set, who has this situation with the BRCA? It happened like few years ago. So you see, you will, Angelina Jolie, you know that she has some operation of the breast because she has some of this. They have in the family the BRCA mutation. So is this characteristic like heterozygosity, right? So if your family has it and you're in breast cancer or in ovarian cancer, or in this case in the retinoblastoma, if they have plus minus, they have chances that it could happen a mutation, and it happened this. And it come up in the newspaper. I don't know if it's, I don't have connections with the shed set, but it said that BRCA was one of the genes that she was having heterozygous. So what happened is that non-doctor, when I was doing my postdoc in Cornell, we work with many people in Memorial Sloan Kettering. And this was like in 10, 15 years ago, they start coming with all these molecular genetic, right, panel. And they say, the surgeons and the people, the oncology, they say, how we are going to do with this information? Because you have this information and you tell the patient, but you cannot guarantee that that person will happen to have a tumor, right? So then nowadays, it's more known that they call the personalized medicine, and they characterize this. And I guess this is a decision of the patient if they want to have a mastectomy or then removal, because they have consideration that maybe will have some mutation that will lead to a tumor. So in this case, that was an example that if some of you hear about that, it was reading that was BRCA, one of the genes. So this is a concrete example where if you have a somatic retinal cell, right, that has a heterozygosity, if one allele is positive, one allele is negative, right? It's RB plus minus. So the heredity retinoblastoma, the somatic mutation, it will lead to homozygous cell give to rise to tumor in the retina. Why? Because RB is no more function, right? It has minus minus. So this is when you have it, heredity one, and then in the life of the person has another mutation. In this case, it happens sporadically. So the same person has first one hit, one mutation, so it becomes heterozygous. And then what happens? A second mutation, it will give homozygous cell and they have tumor. So there are two types of has this, one heredity, retinoblastoma, and then the baby already has this, and then it will get one hit in the somatic cell. So the mechanistic for loss of heterozygosity, there are different types. Missegregation of the chromosome, like we talked about before during the mitosis, recombination between chromatids with the mutated allele and the normal allele, or chromosome deletion, including the normal alleles. So these are all the possibilities that you can have loss of heterozygosity. And when it happened, so those are on the point of the molecules of the tumor suppressors. So what happened when the oncogenes? And now we go back to the oncogenes because there are some concepts that I want to see what happened when the oncogenic mutation may involve in different things. One are oncogenic receptors, then could be also in the active signal transducer, or then on the growth inhibitor signal, on the cell signal control. So there are different ways where the oncogene mutation may involve. So when it happened in the receptor, right, the receptor become oncogenic. So what are these different characteristics? One is mutated, the receptor can dimerize in absence of the ligand. In general, all these ligands, the receptor, like for example, EGF is one of the basic, a lot of cancer has also EGF mutations. So what happened, the EGF is epidermal growth factor. Epidermal growth factor receptor has a mutated, and then into that mutated protein is called new oncoprotein. This is characteristic in breast cancer. Another way, in general, these receptors are those ones that they have to dimerize. They might see it, you might see it, it is in basic biology. So they need to dimerize to be able to make the signaling, right? So what happens is that in generally you have a ligand that bind, dimerize, signaling transduction to the nuclear. So in this case, the extracellular, this was the signaling that was dimerized, right? Receptor is mutated. Here is the ligand binding domain is deleted. So they don't have the binding domain, the binding of the ligand. So lead to constitutive active. This is, like I said, the example of the short circuit. You have signaling, even though there is not really, has to be signaling. So that is another case when the deletion of the EGF receptor compared to a special oncoprotein that is called RBB. So these are examples I say because, you know, if somebody has some familiar that has, for example, breast cancer, they do some molecular staining and they stain in these molecules to see if there are some receptor positive or no. Because based on that, they will be able to give some treatment or another treatment. That is why it's important, and I'm bringing to you this concept. Also, sometimes mutations can lead to overproduction of a normal receptor. So this is the example of the HER2 overexpressed in breast cancer. And another thing is abnormal receptor resulting chromosome translocation. This is another thing. What happens is constitutive active signal. We say all of these are gain of function, right? We say that in the previous. Many oncogenes derive from protein involved in transduction of signaling, as I said before, and promoting growth and proliferation. So these are examples, and some of the two graphics are here. For example, you have, this is the HER2, right? That you have binding by glycine, so then you have new oncoprotein. So what happened is that they are some mutation, so they will happen is that they have signaling. So even though you don't have the ligand, right? So the ligand-independent receptor oncoproteins. In this case, it's a new because you have a mutation. You don't know, I don't remember, it's glycine. I think it's valine for glycine, but it doesn't matter. You don't need to memorize that, but you need to see it's a point mutation. One mutation lead that one amino acid changed by another amino acid. So now you have a new protein, it's called new oncoprotein. It should be like this, but it's like that. And in this case, right, so you don't have the ligand part here, right? But then constitutive is get activate the tyrosine kinase, that is the one that's signaling, and it continuously phosphorylating like it is a normal, but in reality it's not like that because it has no the ligand, right? So this is due to different, in this case it's a point mutation that changing the amino acid, this is a deletion, right, for all this area. So some examples are, I say the RAS is very, RAS is a small GTPase. For those that, you know that the GTPase bind to the air and then they convert, they process and then that GDP. So that is a continuously very active molecule in transition, in signaling transduction. And what happened is that any mutation in there, it changed the activity of this GTPase. So, and it's very characteristic in many type of tumors. Then we are going to see the one that it was very special. I mentioned to one that it gives the opportunity that this is an oncogene and generate a new fusion, right? Fusion of two genes, CABL with the BRAC. So what happened is that CBL encodes for some tyrosine kinase, and the BCR is unknown. What is the production of that molecule, right? But what happened is that when you have, we say about the translocation, right? They have some chromosome rearrangement. So what happened is it generates this BRAC-ABL is a new oncoprotein form, right? It's a tetramer with continuous and upregulated activity. So that is, it doesn't matter that is the pathway that follows, but the more important is a new protein. That protein is a fusion protein and it was not in the normal conditions. So here is the graphic of how it happens this. So most likely is diagnostic fusion in the chromosome Philadelphia, right? They come from the diagnosis by detection. When they do it, the genetics, a study of the karyotype, they can detect what is here is a small chromosome they call DER22, that is Philadelphia chromosome. So what happened is that is a loss of function mutation because what happened is a translocation. This is a normal chromosome 9. This is the normal chromosome 22, right? They have this translocation, rearrangement, and then what happened is generating this chromosome, and that chromosome, it will have the sequence to produce this gene that the transcription is this oncoprotein fusion. So this is very characteristic and is happen usually in the chronic leukemia and the acute myelogenous leukemia. So this is one characteristic when something that was not exist, it happened because this rearrangement, and then now you have a new protein that is a fusion protein. And that was very important to introduce the concept that was used to generate this drug. So they study with many, different scientists, they study and they come up with this structure that if you see, this is the molecule, we say the new molecule, and they have area is ATPase activity, right? So what happened is the ATP is there because you need this for them be activated, right? It's an ATPase activity. So what happened is the ATP is hydrolyzed and then is ADP and phosphorylate the other part. This is in the leukemia. So what happened is they designed this drug and they realized that they dock and they can be in the area where the ATP site. So then that drug is located in that area, so then they cannot be phosphorylating, they cannot be signaling, so then they are not going to be the leukemia, the production of the leukemia. And this was having very good result on the clinical. I don't know the last update, if they are needed or not, because sometimes what happens is that the tumor has a resistance, so become resistant, so then they have to give another drug or combination. But then what happened is the important point here is that you have a new molecule, right? A new protein that is a fusion protein, that. And then the very important is how, by the study of the structure of these molecules, that was new. So they studied that and they say the kinase is the area where the ATP sits. So then they use that domain of the kinase and inhibit the enzyme. How? They inhibit with designing this drug that is exactly the treatment that locate in that area, cannot make the transduction of the signal, so then they cannot proliferate the cells for the leukemia. So that is a very unique experience that they come up with this drug that use the structure new that is in this type of tumors. Any questions? So now we go back to the picture of the, let me see, this is to refresh how is there, we say the very important, the retinoblastoma is controlling when it's no proliferative cells, right? So then when it's phosphorylated, this is released. So then it's phosphorylated, this phosphorylate the retinoblastoma and release this active transcription factor, and then be able to continue with the cell cycle and all these S-phase genes that they needed for the synthesis of DNA. Why this is important is because I wanted to bring the point that there are some of these molecules that are important in cell cycle, like for example, there are growth inhibition signaling, right? So some of them is the transforming growth factor, TGF, that what happens is that promote apoptosis. Sometimes there are mutation on this. So then, for example, the P15 is an inhibitor of the cell cycle. Pi is an inhibitor of the proteases. Remember that for the tumor to metastasize, need to secrete proteases, right? So in this case, if there is a mutation in this gene, there are increased proliferation by blocking this P15. And then in the case that if the inhibitor of the proteases is affected, you have increased risk of metastasis by blocking this inhibitor, right? So these are just examples how there are more examples of how a tumor can become malignant. So there are many, as I mentioned before, in general, the inhibitors, the cell cycle inhibitor are named by P16, P15, P21. So the main point of this slide is to show you how there are some regulations of the cycling decay and the cycling. And then this is the same story of the retinoblastoma. So there are many components that they are part of this regulation, that in the cancer, if they are mutated, then it will lead to the dysregulated proliferation. Okay. So then this is a summary of what we said before, right? So we have the cycling decay, and then the phosphorylated inhibitor, the P. So then what happens is that they are released by the retinoblastoma and then promoting the transition between G1 and the S phase. So then if the mitogenic stimulus is removed, then what happens is that there are a dysregulation in different, the CDKs are inhibited by this inhibitor of the cell cycles. So the very common cause of cancer involve mutation leading overproduction of cyclins, or the loss of retinoblastoma, or the loss of the inhibitors. So some of these, what happens is loss of one of these components is sufficient to induce excessive proliferation. Basically, it's a summary to what we were saying in the past several slides, right? So this is to refresh you how the P53, if it's mutated, then what happens is that that DNA damage, P53 is supposed to solve this situation, making the cell arrest, DNA repair, or die by apoptosis. But what happens if you have
mutation P53, nothing happened, and the cells keep going with the DNA damage to the progeny. Any questions about this? Because now we are going to enter in the last part of the class. So what happened is that these are the carcinogens, the carcinogens, right? So there are two different types of carcinogens. This is what I want you to know. Natural or synthetic chemicals that cause cancer DNA by DNA damage. You have two. One is the direct active carcinogen, is because the same carcinogen directly get react with the N or O atoms of the DNA bases. So modifying the base structure and pairing, right? So then that will be changed, the structure of the DNA. What they call is a DNA adduct, because it's kind of like a deformation of the helix. So then it's more difficult for the cells to replicate that DNA, right? So then also another type of carcinogen is indirect carcinogen. What happens in general are no polar chemicals, so they become reactive after they are modified by cellular enzymes. This is in line of what this is how they can be acting, the different chemicals they can do initiation and then promoting in different steps for the mutations. So the example is like, here is one example. So what happens is that when you have a toxin, right, or a chemical, in this case, the carcinogen, the liver has these enzymes, detoxify enzymes, and they will try to clean up this, right? So then what happens is that when they are chemicals, in this case are, if you see the aflatoxin or the aflatoxin epoxide, so the carcinogen bind to the one in the DNA, right? These are different examples. You don't need to memorize, but you need to know that these are examples that they interact directly with the one in the DNA. So these are direct carcinogen, right? What happened with, for example, in this case, I was doing research with this benzoypyrin. Benzoypyrin entered in the cells, then in the organism is being metabolized by the P450s. And the benzoypyrin is one of the chemicals I was studying because the tobacco smoke, right? It's one of the main chemicals in the tobacco smoke. What happened is that the liver enzymes try to detoxify. How they detoxify? If you check here, right? So they try to make it more hydrophilic because it's a polar react. So they want to eliminate by the urine, right? So the enzymes add oxidryl groups. So those oxidryl group, what happened is that they change the structure, and that structure is the one that react with the one in base. So then this is an indirect example, because the benzoypyrin per se doesn't bind to the DNA. But when the enzymes try to detoxify and add the oxidryls, right, to make it more hydrophilic and be able to secrete it by urine, what happens is that is very reactive. So they can be interact with one molecule of the DNA, and then it produces the carcinogenesis. So that is also another example of how this is basically, they say when you burn the food, when they do barbecue, but when they burn with the fire directly touch the meat, you have certain compounds that they can be causing also carcinogenesis, right? Not when you cook the regular cook. People sometimes overcook with the fire directly. That all this dust, black dust that they are in the meat, they say that they have some impact, right? Carcinogens. So basically, that was the example. You have two types of carcinogen. One is direct, one is indirect, because they have processing in the liver. And this is the last slide that we are talking about carcinogenetic mutations in the caretaker gene. As I say, this is a normal function that they have. They prevented or repair the DNA damage, and the DNA damage defect lead to hereditary cancer predisposition. In generally, there are two different types. One is xeroderma pigmentosus and a non-polyposis colorectal cancer. These are the caretaker genes that they are in charge to have everything in place and DNA repair, but if there are mutations there, that cannot happen. And this is a summary of all these topics that we saw it before. So some questions? Are you still okay? So please, I know everybody want to go home, but if you have questions before the exam, send me. I am now very busy also. Right now I have another course in the medical school that was given me 1 week ago, and I start this Monday. So I'm very busy. I was supposed not to be busy till March, but so please email me, and then we can find the time to go over Zoom. I don't like to answer questions by email because sometimes could be misunderstanding, but in Zoom or in person, we can explain. And thank you for being here in all this time. Thank you so much. Good luck.